Good morning, everyone. Welcome to our Global Symposium series with Dr. Rebecca Gilbert, speaking for us today on pioneering Parkinson's research from the American Parkinson's Disease Association. My name is Alana Dillon. I'm an Education and Support Services Coordinator at Parkinson Society BC, and I'll be facilitating our webinar today. Before I introduce Dr. Gilbert, I'd like to go through a few housekeeping items. First, I do want to let you know that we are recording this webinar, and it, along with the presentation slides, will be available on our website. Secondly, you will see a chat icon at the bottom of your screen. You can click on that to open it up, and then feel free to use this chat box during the webinar to type any comments or questions that you have for Dr. Gilbert, and she will address the questions at the end of the webinar. And finally, to optimize your experience with this webinar, please do ensure that you adjust your speaker volume both on your computer as well as on your actual speakers so that you can hear the webinar well. It's my pleasure to now introduce Dr. Rebecca Gilbert. Rebecca Gilbert joined the American Parkinson Disease Association in 2018, bringing a wealth of practical experience in diagnosing and treating Parkinson's disease, as well as a strong background in fundamental and clinical science to the team. She is responsible for overseeing the American Parkinson Disease Association's research portfolio in conjunction with APDA's Scientific Advisory Board, and also provides medical and clinical expertise to support APDA programming as well as print and web content. Dr. Gilbert received her medical degree at Weill Medical College of Cornell University and PhD in cell biology and genetics at the Weill Graduate School of Medical Sciences. She then completed her neurology residency training as well as movement disorders fellowship training at Columbia Presbyterian Medical Center. She continues to maintain a limited schedule of patients 1 day a week through Bellevue Hospital Center. Dr. Gilbert, welcome, and thank you for being here today to share your expertise. I will now pass it over to you.
Thank you so much, Alana, for having me, and I look forward to sharing APDA's research agenda and accomplishments. So I'm going to open up my presentation. So I have been asked to speak with you today about American Parkinson Disease Association, our research, how we envision our research, and what we have done in the past to move forward this extremely important topic of Parkinson's disease research, getting to new treatments and eventually hoping getting to a cure. And I know everyone on this webinar today would love to see that happen. So let me start by introducing American Parkinson Disease Association to you. Here is our mission statement, and I'm sure this will resonate with you. Every day, we provide the support, education, and research that will help everyone impacted by Parkinson's disease live life to the fullest. And so we have a three-pronged mission, which includes support services, education about Parkinson's disease to people with Parkinson's disease and their loved ones, as well as funding research to move the field forward. In terms of programs that we provide, we are an organization that exists in a local form in many parts of the United States. We have chapters and information on referral centers in multiple states and locations. And through that network, we provide information and referral, so people can call up and get information about therapists, physical therapists, speech therapists, movement disorder specialists, etc. They can also access support groups and educational symposium on the local level, as well as exercise and fitness classes on the local level as well. Now, of course, during COVID, we, as well as you, pivoted to an online platform, and we provided online programming in the form of education, support, and wellness programming throughout the pandemic, and we continue to do so as well. In addition to our local services, we have many services that can be accessed via our website, and this can be accessed by anyone around the globe. And these include publications, webinars, and a variety of other online resources. In terms of our research, so the third arm of our mission, we are very committed to giving funds to Parkinson's disease researchers that would not have ordinarily received funding. And that's really where we feel our niche is. We have 4 categories of research funding that we give to individuals who are performing Parkinson's disease research. The first category is our George C. Kotzias Fellowship. And this is a very prestigious award that's given to a physician scientist, so somebody that sees patients and does research. It's a 3-year fellowship, and it really is designed to launch a career. Somebody who may just finish training, wants to get their lab going, but needs that seed money. They need to be able to go to their department chair and say, I have gotten funding. Let me have an opportunity here. And that's where we see where our funding really, really has impact. We also fund postdoctoral fellows. These are people who just finished their PhD and are going to work in another person's lab, but they need some money to, again, make themselves worthwhile and make themselves a good candidate to work in someone else's lab, and they need that initial seed funding. We have a research grant category, which again gives seed money to somebody who may be at an assistant professor level. So they just finished their postdoctoral fellowship. They haven't gotten their full lab together, but they need, again, that little bit of money to get themselves started, and that's where our research grants come in. And we have a 4th type of grant, which we initiated a couple of years ago, which we entitled our Diversity in Parkinson's Disease Research Grant. And this is a grant that's specific to someone studying Parkinson's disease in a community that has previously been understudied. And this could include women, rural populations, populations of various ethnicities, native populations, populations that sort of have escaped traditional study. And we feel that this is vital because if we don't understand Parkinson's disease in all its forms, then we're going to miss out in fully understanding the condition in everyone. And so we are committed to that type of grant as well. We also fund centers for advanced research, and this is money that we give to an institution that is performing Parkinson's disease research, has a very advanced program going, but may have some holes in their funding. They may have somebody that's come aboard doesn't have funding yet, or they may have a trainee that they would like to continue funding, but they don't have that sort of stopgap money. And that's where we feel our impact can be the greatest. And so now that I've talked about which individuals and which institutions can have access to funding from APDA, I want to talk about what APDA has funded and why that is so critical. So let's take a step back and really think about what Parkinson's disease research entails and how we can get to the ultimate goal of devising new treatments and new approaches to really conquering this disease once and for all. So as you know, in order to create a new medication that people can take, it requires years and years of what we call preclinical research. So research in animal models, in cell culture, that really works out and defines the mechanism of what is going on a really molecular level before anything can happen in people. And this process is extremely lengthy and may in the end not lead to a chemical or a new product that can be used or taken by people. And we have to be open to those failures because not everything is going to succeed and make it through. In fact, the vast majority of ideas do not make it through. And that's the nature of our very, very complicated body. It is so complicated that working out the details just requires a lot of effort and time. And so funding those early projects, the projects that are looking at the molecular level of what's going on in the cell, but are looking at it in a culture dish, looking at the cells in a little glass dish, or looking at an animal model of Parkinson's disease, a mouse that has been engineered to have some of the clinical features of Parkinson's disease, and studying them to see why Parkinson's develops and what we can do to halt the progression. All that needs to happen before anyone can start thinking about how this may affect people. And so APDA funds all that process, from the preclinical all the way to the clinical research, which is research that involves people. Now, having explained that, what we call the pipeline of research, which is a very long and elaborate pipeline and fraught with failure, I want to talk about 6 really exciting themes in Parkinson's disease research. And APDA has funded projects in each of these 6 categories. Now, this isn't to say that there aren't many other themes in many other categories, but I'm going to focus on these 6. And I'm going to talk about what these 6 themes are. I'm going to talk about different projects that APDA is currently funding in some of these categories. And then I'm going to talk about research that APDA funded in the past in these categories. And I hope that this gives a glimpse of the exciting topics and movements that are happening within the Parkinson's disease research world. So the first category of Parkinson's disease research that is very exciting, and APDA and many others are funding, is the concept of the abnormal accumulation of alpha-synuclein, the top left picture. Now, what does that mean? As you may know, Parkinson's disease is characterized by a very specific blob, that pink blob within the cell that you see, that pink cell. There's the outlined pink round entity. That is what is called the Lewy body. Inside the Lewy body, there is an abnormal accumulation of a very specific protein called alpha-synuclein. Now, we know that this protein accumulates abnormally. It forms this big mess of a pink thing. When it's dyed correctly, it looks pink. And we think that this abnormal accumulation affects the health of the cell and causes the cell eventually to die. So we think that if we stopped that accumulation or broke up the accumulation, we could have a real impact on Parkinson's disease. And so a lot of research is dedicated to understanding why alpha-synuclein accumulates and figuring out how to make it not accumulate. And so that's theme number 1. Theme number 2, the role of other cell types in Parkinson's disease. So you may know that the brain is made up of billions of nerve cells or neurons. And you would think that that would be where the action is. The nerve cells, they talk to each other. And when that doesn't go right and the nerves die, that causes some of the nerves, specific type of nerve die, that causes Parkinson's. But it turns out that the cell has billions of other cells. The brain has billions of other cells as well that are not neurons. And some of these cells are called glial cells. These are supportive cells in many ways. They may provide support to the nerves. They may provide nutrition to the nerves, protection to the nerves. They may inflame the nerves. They may do all sorts of things to the nerves. And we have been ignoring these cells almost entirely in our Parkinson's disease research. So understanding what these other cells do in Parkinson's disease has become a really exciting avenue of Parkinson's research. Third, brain circuitry. So we have all these nerve cells and all these glial cells. The nerve cells, in order to be productive, they need to talk to each other. And they talk to each other in circuits, much like wires communicate or electrons communicate with each other by being passed down a wire. The message that one nerve has passes down one nerve and then speaks to the next. And that's how the nerves communicate. Now, the brain circuitry in general is extraordinarily complex. When you think about the number of nerve cells in the brain, and it's something like 80 billion, that's just an overwhelming number to think about. But on top of that, another layer of complexity is how those nerves talk to each other, and they are intertwined in absolutely billions and billions of ways. And when some of these brain circuits go awry, that can be a huge problem within Parkinson's disease. So making the brain circuitry more normal, or normalizing brain circuitry that has gone awry in Parkinson's, is a huge and exciting avenue to pursue in Parkinson's research. What about the role of exercise? As I'm sure many of you know, exercise is touted right, left, and center as extraordinarily important for Parkinson's, and that is true. But why? What does exercise do to the brain in a brain that's generally aging or a brain that has Parkinson's disease, what does exercise do to make Parkinson's disease better? So understanding the role of exercise is critical. What about genetics? We all know that we all carry the genes from our parents. We've inherited chromosomes and their DNA from our parents. And we know that our propensity to developing different medical conditions is going to be, in general, a combination of what we inherited and our environmental exposures. So understanding how specific genes may contribute to Parkinson's disease research, or Parkinson's disease risk, is a very hot topic these days. And further understanding exactly what those DNA changes do and why, and can we ameliorate them? And finally, the sixth topic is digital biomarkers. And so what does that mean? It means that if you have a device that you wear, a watch, a phone perhaps, some other wearable device that may be on your waist or on your wrist, it may capture information about your movements that can help maybe diagnose Parkinson's disease or follow trends in your Parkinson's disease. And trying to extract that data and using digital biomarkers to fully understand how Parkinson's disease is manifest in movements that we can record. That's another really exciting topic in PD research. So those are the 6 topics I'm going to focus on today. Sorry. Oh my. I lost control of my slides here. Okay. What we're going to do now is look at some of the specific APDA projects that correlate to these specific 6 themes that we talked about. And here are pictures of our current APDA researchers. First, we're going to talk about Parkinson's disease and alpha-synuclein, that first theme that I mentioned. And we're going to talk about Dr. Sunilkumar, who is studying in the University of Denver. He has a lab there. And his goal is to identify the part of alpha-synuclein that is important for abnormal accumulation of the alpha-synuclein. So we know that the alpha-synuclein sticks together, but which part of alpha-synuclein sticks to which other part of alpha-synuclein to form that clump? If we knew, then maybe we could block it. And so that's what he's working on. Very exciting project. Now let's talk about the theme, the second theme I mentioned. So the other cells in the brain that may be contributing to Parkinson's disease problems besides nerve cells. And as I mentioned, the brain is composed of many different types of cells, not just nerve cells. And we need to capitalize on that to fully understand Parkinson's disease. And now let's meet Dr. Abby Olson. Once again, I'm having trouble. Dr. Abby Olson works at the Brigham and Women's Hospital in Boston, Massachusetts, and she is studying the role of glial cells in Parkinson's disease. And what she's doing is she is manipulating the genetics of a fruit fly model of Parkinson's disease. And she is trying to discover the specific genes related to the glial cells and how they may drive Parkinson's disease in this very primitive Parkinson's disease model. And using this very simplified model, this fruit fly brain is really tiny, but it can unlock secrets that are just much harder to interpret in a larger brain. And she is looking at how glial cells may contribute to therapies for Parkinson's disease. Now, what about Parkinson's disease and brain circuitry? What are we funding in that realm? So let's talk a little bit about deep brain stimulation, because that is a therapy you may be familiar with that is extremely important in understanding circuitry of the brain. You may know that deep brain stimulation is a neurosurgical procedure, and it involves putting electrodes or wires deep within the brain, and it delivers electrical impulses to deep brain structures in order to improve Parkinson's symptoms. So basically what deep brain stimulation is doing is manipulating those circuits I talked about to try to make the circuitry that's abnormal in Parkinson's disease act more like it should. And these electrodes are connected to what's called an implantable pulse generator, or an IPG. It sits underneath the skin in the chest. And this can be programmed remotely by a programmer that a doctor holds. And it can be programmed with various variables of the electricity, how wide the electrical pulse is, how tall the electrical pulse is, how fast the electrical pulses come, exactly where the electrical impulse is placed, etc. So all those variables contribute to how the circuitry is manipulated. We have a researcher who works at Emory University in Atlanta, Georgia. His name is Dr. Enrico Opri, and he is looking to make deep brain stimulation, or make this manipulation of brain circuitry, even better. And how is he planning on doing that? So he has identified a brain signal, which he has called DLEP, or deep brain stimulation local evoked potential. And this he has discovered, it's a brain signal that seems to be capturable. He can capture it on his recording devices. And it is present even when the brain is under deep anesthesia. So you may know that deep brain stimulation typically is done when a person isn't under deep anesthesia. They're more awake because you need to really hear where the electrode is in the brain, and you have certain signals that you can detect. And those signals go quiet when somebody's under deep anesthesia. And sometimes people are nervous about having deep brain stimulation when they're not fully asleep. Now, there is one way around it these days, which is MRI guidance. So the electrode is, patients are put to sleep, all their signals go quiet, and the electrode advancement is done using visualization, using an MRI. However, that isn't ideal because you can't really hear where you are. You can just see. So you have a visual guidance, but you don't have a functional guidance about what the cells are doing. So Dr. Opri has discovered this signal, which can be detected even under deep anesthesia. And so you can hear where the electrode is, even under deep anesthesia. And he's hoping with this signal to further refine the technique and make it possible to do deep brain stimulation in a very accurate way, even under deep anesthesia. So really excited about that project. What about Parkinson's disease and exercise? And what is APDA funding there? Now I want to talk about this project. This is not a project APDA funded, but it's one of my favorite projects to talk about because I really think it brings home the message of exercise and what exercise may do. So in this project, or in this study, the researchers had rats that ran on two different kinds of wheels, of exercise wheels. On the left, there was an exercise wheel that was called a skilled exercise wheel. And so different slats from the exercise wheel were removed in a relatively random fashion. And this required the rat to learn where to put their paws. So they weren't able to run that fast, but they had to do something with some cognition involved in order to walk carefully on this wheel. The other wheel was a more traditional wheel. The slats were evenly located. The rats knew very well how to run on this wheel, and they could run really fast. So we had this cognitive exercise and we had this aerobic exercise, and then the rats were compared. And it turns out the rats with the cognitive exercise, they needed to go slower, but they exercised with a cognitive component. Their brain actually looked a lot more active than those who exercised just aerobically. And I think this is super interesting because it really teaches us how important that cognitive piece is to combine with exercise, and why it seems that the push in Parkinson's exercise often involves learning a new motor skill. That may be boxing, tai chi, karate, tennis, whatever it may be. Along those lines, I want to highlight another really exciting study. And this was sponsored not by APDA, but by NIH, National Institutes of Health. It was called the SPARK study. And it actually looked at early Parkinson's patients who were not on medication yet, and they were either enrolled in high intensity treadmill exercise or moderate intensity treadmill exercise, or they weren't enrolled in the study, they were the waitlist control, so they didn't do anything. And these were people who had really mild disease and didn't really need any major interventions from a medicine point of view. And they just exercised. And after 6 months, the high intensity group did much better than the waitlist group. And this really further pushes our knowledge and our understanding that moving, especially high intensity moving, is really, really important for people with PD, whether, and as early as you can possibly start to do this for yourself, the better. Again, why? What is going on in exercise that is so incredibly vital for Parkinson's? So we have that cognitive piece to it. We have the high intensity piece, combination, whatever it may be. What exactly is happening in the body when we do these exercise regimens? And we're very excited to be funding Dr. Constanza Cortez from the University of Alabama in Birmingham, Alabama. And what she's doing is she's looking at, as I just stated, the molecular underpinnings of why exercise is what we call neuroprotective, or doesn't allow that progression to be as aggressive. And she's looking at a mouse model of Parkinson's disease. And what she's hoping eventually to show is that when one mouse exercises, there is a product that is produced by possibly the muscles of these mice that can actually be captured and purified and given to another mouse. There might be exercise in a bottle coming where we can harness the power of exercise in a chemical and use that to help our brain. So very exciting stuff. So I mentioned a few of our projects that we're funding now and some of the themes that those projects correlate with. And we have others, we have many others. I didn't want to go through each one because there isn't enough time to really give each one the right attention. And I also wanted to make sure that we looked back, we looked at the past of APDA funding, because it's one thing to say, you know, I think this project's going to take us somewhere. Let's fund this. There's another thing a few years later to look back and say we were right. That funding did result in something positive and substantial for the field. And so at the same time that APDA is excited about our future and what we're actually funding now and what that's going to lead to, we always need to look back and do our due diligence about what we already funded. Did we do a good job? Did we select good projects that led to good results? And one of the ways to do that is to look at the scientific papers that were published from the work that we funded. And here are a selection of some of the papers in our past that highlights the work that we funded and what the results, the scientific results were. So let's go through some of those projects and look at the results that APDA funding led to. So let's look at our first category, alpha-synuclein and Parkinson's disease. And we funded Dr. Vivek Uni. He was one of our COTSIUS fellows. You may recall that's the 3-year fellowship where we launched the career of a physician scientist. Dr. Uni is both a neurologist and a researcher. He works at OHSU in Portland, Oregon. And he studied the molecular mechanisms of alpha-synuclein. So you may assume that since alpha-synuclein is so important in Parkinson's disease, it forms these globs, and we think that's the key feature that causes the problem in Parkinson's neurons, you would think we would know what alpha-synuclein does normally in normal people, because it has a function every day in our brains of people who don't have Parkinson's. It just kind of, but what is it supposed to do? We need to know that for sure, but we don't. So Dr. Uni looked at that very, very important question. And he found a really unique function of alpha-synuclein. And this may mean that alpha-synuclein does a bunch of things. What he discovered is that alpha-synuclein plays a role in repairing damaged DNA. And so what you may know is that our genes, made up of our DNA, needs to divide in order to form another cell. And sometimes when that happens, the DNA gets a little damaged, and there's a whole system involved in our cells to repair that damage. Because if there is damage that isn't repaired, you may create new mutations that can lead to problems. It turns out he found that alpha-synuclein is vital in repairing damaged DNA. So you can imagine that if alpha-synuclein abnormally accumulates, it no longer is repairing the DNA, and that can lead to problems. And maybe that's part of why Parkinson's happen. So that was really exciting research to be part of. What about the Parkinson's disease and genetics category? we funded a scientist called Karen Neutemans, and she works in the University of Miami, Florida. And she was looking at the genetic components or genetic specificity of the Parkinson's disease community in Hispanics in Miami. And as I mentioned before, it is really vital to understand Parkinson's disease in all its manifestations, because if we only focus on one population, we literally will be missing not just the important features of Parkinson's in other communities, but the important features in all communities. So you can imagine that there may be one ethnicity where one genetic mutation is very strongly represented, but in fact, that's only a good population to identify that mutation. But that mutation is really important in all populations. We just would never have noticed it if we weren't studying that specific population. And we know this to be the case in specific DNA changes. You may have heard of the protein LRRK2. That was discovered because a certain group in Northern Africa had a really high percentage of Parkinson's, and LRRK2 was discovered in them. It turns out that protein is extremely important in all people with Parkinson's. So really spreading our understanding of Parkinson's is absolutely critical for everyone. And so Dr. Neutemans is focused on the Hispanic population of Miami, and she in fact discovered a new mutation in LRRK2, which we didn't know about before. It was never highlighted before in any other population. Turned out to be specific in her population, but now we'll know to look for it in everyone. And so her work is extremely critical. Another aspect of genetics that we cannot miss discussing is the differences in genetics between men and women. Of course, men have a different chromosome, the Y chromosome. So are there differences in Parkinson's disease between men and women? And we funded a scientist called Dr. Elizabeth Disbrow, and she works in Shreveport, Louisiana, Louisiana State University Health Sciences Center. And she studied the cognitive differences in Parkinson's disease between men and women. And she discovered that men with Parkinson's had significantly greater executive and processing speed impairments. So those are certain cognitive domains that were worse in men than in women. And she found that these differences were not related to any other variable or any other measure of disease severity. It did seem to be very related to gender. And figuring out why men have a slightly different cognitive profile than women is her next thing on her agenda. In terms of Parkinson's disease and digital biomarkers, we funded a scientist at the University of Michigan called Dr. Guan, and she looked at the potential biomarkers that can be extracted from a mobile phone, from a cell phone. And there are embedded within a cell phone, within every cell phone, what we call accelerometers and gyrometers, which are ways to, which are necessary when we manipulate our cell phone in different ways and the picture changes from landscape to portrait, or we move our cell phones around and the cell phone is aware of that. So there's a lot of data that the cell phone is programmed to do. And she harnessed all that information that the cell phone generates or that the cell phone records, and was able to extract a biomarker based on how people move their cell phone and how people use their cell phone to identify Parkinson's disease. And this is actually extremely interesting, and it goes to the potential for our digital world to diagnose Parkinson's and then also to follow Parkinson's. Maybe this is a good way to see if a clinical trial drug works, because we know how the person's movements were based on their cell phone movements at the beginning of the trial and then at the end of the trial, and has that improved? And it would be an objective way to measure Parkinson's disease movement. And this is something that has sort of escaped us to a certain degree. We now are measuring people's Parkinson's disease based on what we call the United Parkinson's Disease Rating Scale, UPDRS, which is a way of determining how people are doing, but it is really very subjective, and it really varies varies somewhat from different examiner to different examiner. And using a digital biomarker may be a much more objective way to capture whether people with Parkinson's movements have improved And that could be super important in all sorts of ways, including clinical trials. What about Parkinson's disease and brain circuitry? We were very excited to have funded Dr. Mallory Hacker, who works at the Vanderbilt University Medical Center in Nashville, Tennessee. Now, you may know that deep brain stimulation, we talked about that a little earlier, is a procedure that's typically performed in more moderate to advanced stages of Parkinson's. It's not usually done early in the disease. But Dr. Hacker followed a cohort, a group of people with Parkinson's who had deep brain stimulation performed at very early stages. And she followed these people over time, and APDA funded the 5-year check-in to see how these people were doing. And she was able to report that participants in that early deep brain stimulation group, along with the medical therapy they received, did better than those who only received medical therapy, and that was at the 5-year mark. And so very excited to see the next stage, the 10-year mark, and how these people did. I want to highlight again that one of our prime goals as an organization that has programmatic education and research as our 3-pronged mission, I want to highlight, it's really important to us that we fund research both in the past and in the future that's going to lead us to helping people with Parkinson's. Obviously, that is the ultimate goal. But on the way, we very much want to take the time and the energy to explain the research that's happening to the people with Parkinson's. And we want to embed our understanding of research into our programs, into our education. And the reason for that is you deserve to know, first of all, what the funding is going toward and what the funding has done, because it has real life impacts on you. And we are very mindful of that. And we want to make sure that you understand what the money is being spent on and why. And so interpreting the results of our research, explaining our research to people with Parkinson's disease, and we do this in various ways. So last year we had a virtual research roundtable. You could see that on the left. We had 4 of our researchers discuss their work and then answer questions from the audience. And we highlight our researchers constantly on our website with interviews with our researchers, discussions of past researchers and what they've accomplished, and just really trying to drive home the message that we need to make this research understandable to the people who have Parkinson's disease. On that note, I want to highlight our important website again, apdaparkinson.org/research. And I want to invite you to explore our website and find out even more about research on our website and through any of our social media channels. And here are our social media channels. So on that note, I want to thank you very much for your attention, and I am happy to take any questions. Let's see if there are any questions. Stop sharing. Okay, so we have a question from Bob DeWitt. Among the research outputs on exercise that you funded, are we getting closer to more prescriptive exercise prescriptions? Okay, that's a great question. And what I'm going to editorialize your question a little bit to say, what you're asking is, what is the best exercise for someone with Parkinson's disease? So we talk a lot about exercise, but I need to know, says Bob, exactly what I need to do to make my Parkinson's better. And yes, I think we are getting a little closer to that prescription, exactly what you need to do. So let me say a few things about exactly what you need to do. So the first thing is that the CDC, which is the Center for Disease Control in the United States, has put out guidelines, and they have recommended 150 minutes of moderate exercise or 75 minutes of intensive exercise for all older adults, along with weight-bearing exercise. Now, what does moderate exercise mean, and what does intensive exercise mean? So moderate exercise means that when you are exercising, you can carry on a conversation. When you are doing intensive exercise, you cannot carry on a conversation. That's sort of a good cheat sheet. And if you do reach that, then you are following the guidelines for all older adults. In terms of additions for Parkinson's disease, probably the two additional features of Parkinson's would be stretching and balance exercise. So you'd have all four. You'd have aerobic, you'd have strengthening, you'd have balance, and then you'd have stretching. And if you can get all four in, then you are sort of closer to the ideal. And the next element of the prescription would be that cognitive piece, which I mentioned. So if you can put all that into a regimen, that would sort of be the ideal Parkinson's regimen today. And that would be how I would answer your question. Practically, if you're sitting there listening to that very long description about what one should do in terms of exercise, that may seem very overwhelming. And so that is not the goal. The goal is not to overwhelm yourself and do nothing. The goal is to take on a task that you know you'll do and that you will enjoy, and that you will do regularly, and that you will do every day. Because if you say 5 times a week, it doesn't happen. You need to do it 7 times a week, and you need to do something you can do. So you want to break it down and try to follow as many of those elements that I mentioned. I hope that answers the question a little bit. We have a question from Dean. Does high intensity exercise mean running is better for me than simply walking? Okay, so probably yes. We don't want to, again, discourage people and say, if you're not doing aerobic activity, then it's useless to sit down. Of course, that isn't true. But having that aerobic component where you're actually getting your heart rate going is probably really important. And so is running specifically good? That may not be ideal because of damage to knees and whatnot. It may be a treadmill, it may be an elliptical, it may be some other way of getting the high intensity exercise. But in all likelihood, getting your heart rate up is part of the equation. I should mention here that it's really important to get the advice of your doctor in any of this, because you may have specific elements that you can't do because of some other medical problem you have, a cardiac problem, whatnot. So don't do anything on your own. Discuss all these changes with your doctor. And what I would do is ask your doctor for a referral to a physical therapist. Discuss all this with the physical therapist so that you have your tailored plan that makes sense for you. Next question from John. Is there any relationship between exercise-produced compounds and BDNF, I think you're talking about, which is a protein in the brain as I understand it? Okay, so what Dr. Constanza Cortez is working on is probably not going to end up being BDNF, but BDNF has been shown to be important in this process. So there are different research agendas or different research ideas about what exercise is doing. There's lots of different theories. They may all be true. And one of those theories is that BDNF is very important and may increase with exercise and then feed the brain in good chemicals. So BDNF is probably an important part as well, but not necessarily what Dr. Cortez is working on. Next question from Janie. Are the 150 minutes daily or weekly? 150 minutes is weekly, and I want to apologize if that was not clear. So that would be, 30 minutes 5 times a week. Again, my personal opinion is that 5 times a week is dangerous because you give yourself 2 days off and you kind of get out of the habit. But it is weekly. Okay. We have a question. I was wondering if you could elaborate on the future possibility of a chemical in a bottle. Sounds very interesting. So here's the thing. This is so nascent that I don't have much more to say. We just started funding this work, and more to come as there are more things to say about it. But the general idea is that there may be something produced in an exercising animal, possibly from the muscles, that is what is neuroprotective and can be harnessed and given to another person who's not doing that exercise. But that's all I can tell you at the moment. All right, next we have a question from Angela. What are your thoughts on photobiomodulation therapy? I do not know enough about photobiomodulation therapy to answer that question. So I apologize, Angela. I will not be able to give you insight. It is true, and this may be what you're referring to, there are different light therapies that are used for various medical issues, and different frequencies of light that potentially are helpful in different conditions. And I have seen papers on blue light, red light, or multi-chromatic light, which is all the different kinds of light mixed into one, which is usually how it is, and whether they can be helpful for Parkinson's. I think there's probably the most data about sleep and Parkinson's. So introducing light in certain parts of the day so that you are able to sleep later. And that certainly may be a very effective way to increase ability to sleep using light therapy. But again, there's probably a lot that we don't know about how light affects us in various medical conditions. So to be continued. So I think I may have answered all the questions that have been asked so far. Let's make sure of that. I think I have. So if we'll wait a couple more minutes, if anyone else has any questions regarding APDA's research, our projects that we've funded in the past, projects that we are funding for the future, or any other questions that I can address now, I will be happy to do so. So, Alana, I don't know if you want to talk with our audience at all. Introduce another potential question.
So it looks like there aren't any more questions. I do remember someone asking me a question in the past. You might be able to touch on this. They were wondering about the role of the am I saying this correctly?
LRRK2?
Yes, K2. Is there, in the role of PD development, is there a test for that? I think they were asking about that.
Yeah, so there is. You can get genetic testing, and LRRK2 would be one of the genes that is tested. You do need to be careful because the different laboratories will test LRRK2 in different ways. One way is to sequence the entire gene. Another way is to test for specific mutations. And so you need to know what test is being done to know exactly what information you've just received. So if you are being tested for a specific mutation and it ends up you don't have that mutation, well, you might have a mutation somewhere else that wasn't captured. But if you get the entire thing sequenced, then it might identify changes that are irrelevant. Like, oh, this looks like a problem, but in fact, it's just a normal variant that maybe few people have, but has no consequence. So there are pros and cons to both ways of doing it. So you just want to know what your testing is doing. And you need to have a conversation with your neurologist about that, and potentially even a genetic counselor who can really give you information about what the test is doing and what the results mean for you. So now a whole bunch of questions came in. Can you comment on later stage alpha-synuclein drugs or vaccines? Great question, Bob. So yes, as I mentioned, alpha-synuclein, huge topic in Parkinson's disease. And that theme that I mentioned does have a whole spectrum of drug pipeline. So there are small molecules that are in clinical trial now. You know, I had that slide that showed preclinical work and clinical work. So there are small molecules being tested in humans that are meant to break up alpha-synuclein accumulation. A bunch of them, there's a whole bunch of them that are in clinical trial now. So that is very exciting. As you mentioned, this concept of vaccines. So one strategy to break up the alpha-synuclein or to make the alpha-synuclein not abnormally accumulate is giving an injection of either alpha-synuclein, which then kicks the body to making antibodies against alpha-synuclein, which then is meant to keep the alpha-synuclein from sticking to each other, because the antibody will block the alpha-synuclein and help sort of protect it from finding another molecule to accumulate with. So vaccine strategies are in clinical trial as well. There's a bunch of companies working on this strategy. And that's in addition to these small molecules, something that you would just take orally to break up the alpha-synuclein. So there are, a whole bunch of clinical trials now that we're going to see the results roll in over the next few years to see if this is going to be a drug that we can take that's going to be neuroprotective. So that's very exciting. We have a question from Dean. I have had DBS on both sides. It's helping my tremor, but it's affecting my speech. So this is definitely something reported. Not everybody has this problem, but it has been reported. The way to address it typically is to try to find the DBS settings that don't do this, that cause the good things from DBS and don't cause the side effects. And there are, so it used to be that one company, at least in the United States, one company made all the DBS. There was no competition at all. In the last 5 years, 2 other companies entered the market, forcing the first company to really up their game. So we have 3 really exciting markets, 3 exciting products, and they all have much more advanced programming options than ever before. And so really tailoring that programming is something that can be done now. And so that may be something to explore. Is there any settings that don't do that to your speech? That would be what I would say. Deborah put in a question. I'm interested in knowing why a person who exercises 2 to 3 times a day, hours per day, has not seen a slowing in the progression. So I'm not going to argue. Yes, this is an inexorable disease that progresses no matter what we do to a certain degree. So the goal with exercise is not going to be to stop progression, unfortunately. And in addition, there are some people whose progression is more aggressive than others. There's a lot of variability to progression, and there's a lot we don't know about why one person progresses worse than another. And so our general understanding about what exercise does is that it slows down the progression, it doesn't stop the progression. And in certain people, it may not seem to be making a difference at all. And there's going to be a huge variability there. So I don't want to, I think you're making a very, very, very critical point for everybody to listen to, that we tend to focus on the positives and the importance of exercise and what it can do, but we cannot ignore or deny the experience of people who don't find exercise as helpful as we're saying it is, because of that variability. So thank you for pointing that out to us. David had a question. Do you think there will be a new drug that will be better than levodopa, as that is the gold standard? So it is important to note that Parkinson's disease motor symptoms are due to the death of dopamine neurons, which means that some way or other, you need to have dopamine to normalize movement. Just so happens that that fact was discovered a long time ago, but it does not mean that dopamine is not important because it was discovered a long time ago. We need to replace dopamine. That I think won't change. Will there be a better way to replace dopamine than giving levodopa? Well, certainly researchers are working on that. And we think that a better way to give dopamine is to give it in a more even way. So instead of giving bursts of levodopa 4 times a day, to deliver it through there's a system where you can get it through your GI tract. In the United States, it's called Duopa. Or there's a subcutaneous pump that's being developed where levodopa is going to be introduced slowly instead of in bursts. There are drugs, different formulations where the drug is released in a more slow manner. So I think those are going to be very important to introduce levodopa in a more natural and brain-friendly way. But I don't think we're ever gonna be able to escape dopamine, because that's one of the essential problems in Parkinson's. Okay, we have a question from Larry and Jillian. I'm surprised that a large enough early stage PD were not receiving any drug treatments. I've always heard that people are usually diagnosed at the later stages. How are these people identified? Okay, great question. There are many people identified early who don't need medication. And that may vary based on where you live. It could be that maybe people who have access to big medical centers are diagnosed early. I don't have the data on that, but yes, certainly, you know, the push is, of course, to, if you get regular medical care, if your general doctor has familiarity with Parkinson's disease, if your family has some familiarity with Parkinson's disease, they may be able to identify the signs at this early stage where medicine is not necessary. So that definitely happens. And I agree with you, not for everyone. But if we do determine on these various studies how vital exercise is in those early stages, then it will become even more important for everyone to be diagnosed at that early stage so that we can slow down the disease. Because it could be that you really need to identify it that early in order to make a difference, and that no drug can be neuroprotective after a certain point. So as soon as we get a drug available that's neuroprotective, that's going to slow down the disease, we're going to need to identify people earlier and earlier to make sure that drug is as useful as possible. Let's see. To expand more on not seeing differences in progression of PD after doing 2 to 3 hours, could doing an exercise that is not novel, as in person not learning, not slow down? I would be hesitant to say that. That sort of gets back to that first question of like, what is the ideal, and you know, suggesting that if you don't do the ideal, then that explains why you didn't progress. You know, that explains why you did progress, and if had you only done the ideal, you wouldn't have. I don't think it's as simple as that. I think we can come up with what we hope is the ideal Parkinson's disease exercise regimen, and there will still be people who defy that. People who do everything right, eat their Mediterranean diet, exercise 2 to 3 times a day, do all the right components, and yet the disease is taking its toll and we can't get it to stop. And that, I think we have to just accept that for now. We have to accept that reality until we have more neuroprotective agents in place. And this is the case, unfortunately, with every disease. Disease is variable. Any disease, and so you're always going to have the outliers, and you're always going to have the worse and the better within a category, and we do have to be very mindful of that when we're giving talks to a large group of people. I do want to mention, back to Larry and Jillian's question, so I did mention this SPARKS trial where it looked at 128 people in early stage. I want to mention, super important, that the SPARKS-3 trial is going on right now in the United States, which is a phase 3 trial, a much larger trial of this same idea. Increased or high intensity exercise in early PD before medications are started. And it's going to be in hundreds of people, many sites. And it's going to collect the data that we need to just absolutely prove once and for all about the neuroprotective effects of Parkinson's. So we have a question from John. Do you have any ongoing research studying the role of the microbiome? So the microbiome, as you may or may not know, for those listening, is the collective bacteria, virus, various other cellular organisms that live in our gut, typically in our gut, but live on our skin and in our bodies, and affects our health in ways that we haven't really fully appreciated. So in a similar way that I was saying that there are 80 billion nerves in the brain, isn't that enough to study? But in fact, there are other nerve cells we've been ignoring. So in a similar way, we have this gut, lots to understand in the gut, but in fact, there are billions and billions of bacteria in there affecting it too that we've been ignoring. And what does that what does that tell us about Parkinson's or other medical conditions? And people are starting to study this. Is there a specific microbiome for people with Parkinson's? Does it look different? Are there specific bacteria that are under or overrepresented? And what does that do to the absorption of medication, to developing the disease to begin with? Because one of the theories is that Parkinson's may start in the gut. And so does the microbiome play a role in that? So that's a huge theme. You know, I mentioned the 6 themes. Microbiome would absolutely be a 7th theme. And APDA has funded many interesting projects about the microbiome. I just didn't have time to mention all of them. You can certainly find more information about the microbiome projects that we fund at apdaparkinson.org/research. So thank you for highlighting that other very, very important theme in Parkinson's disease research. Some great questions. Thank you so much.
Okay. Whoops, sorry about that. Thought I was unmuted. I think those are all of our questions, Dr. Gilbert. And I want to thank you very much for speaking for us today and sharing your expertise. That was a very interesting and informative presentation. And hopefully you can join us in the future and update us on all of these exciting new projects. And thank you so much, everyone, for attending and all of your great questions. And we will see you at our next webinar. Thanks so much. Bye now.
Bye-bye.